Hi, and welcome to the screencast on Chapter 4, Tissues. I'm going to try using this little picture of me thing down there in the corner. Some people seem to think it makes it a little bit more interactive. So I'll be there if you think it's distracting. I can take it off later. But I'll be facing this way because that's where the big monitor is. So we are talking about tissues today. The first thing you want to know is that, as this shows you right here, there are four major tissue types in the body. This means that any little chunk of tissue that you cut off of somebody can be classified or can be put into one of these four categories. And you'll see as we go over them that they all do actually have, there's a rhyme or reason to how the categories are put together, and they're not structured such that we can make it look simple and have only these four categories. So there are actual reasons why things are grouped into the different categories. First up, we are going to talk about epithelial tissue, which we started in lab already. That's skin is one epithelium, and then anywhere else in the body there is a surface, whether it's an exterior surface like the skin or an interior surface like the lining of the GI tract, the inside of a blood vessel, the inside of a sweat gland. Any little hollow portion is going to have an epithelium lining it. Now, these epithelium tend to have certain characteristics in common. We'll go over the first two as we look at the next picture here. So that first characteristic says that they have polarity, which just means they have an apical surface and a basal surface. So if you look here, this is an epithelium. This one happens to be many cell layers thick, so we call it a stratified epithelium. But it's got cells on the bottom, cells on the top, so a basal surface and an apical surface. Even with this single layer thick simple epithelium, there is still a basal surface and an apical surface, and you can see that there's a distinct difference between the two, the basal surface being connected to this pink stuff underneath it, which you are going to find is connective tissue, and then the apical surface being what I refer to as the free edge. That's going to be either the surface of your skin or what comes into contact with whatever is in your digestive tract. Whoops. And then the other thing you want to remember is that the epithelia are composed of, as this says, closely packed cells. And here I cut and pasted this picture in from somewhere else to give you an idea what I'm talking about. So you want to remember that cells tend to stain dark because they have a nucleus in the middle of them, and that nucleus has lots of DNA in it, so it absorbs a lot of stain, which absorbs a lot of light. So under the microscope, those tissues that are most highly cellular tend to be darkest. So here you have skin. All this pinkish purple stuff down here of multiple different textures is different kinds of connective tissue, which connective tissue is some cells, but then some proteins that exist outside of the cells, and that doesn't stain as dark. When you look at the epidermis, which is an example of an epithelium, you can see how much darker it is than the connective tissue underneath it. So generally speaking, whenever you're looking at an organ, the darkest layers of the organ are going to be the different places in the organ where you can find the epithelia. So another characteristic of epithelia is that they tend to be supported by a connective tissue layer called lamina propria, under which you will find what is called a basement membrane. Lamina propria we'll talk about a little bit when we get to the right lab for lamina propria. I believe that is going to be muscle cells this semester, or muscle tissue, but you should know that it's actually in a lot of tissues anywhere you find an epithelium and in many other places in the body. The other thing I wanted to point out here is this basement membrane to give you an idea of what that means. So here we have some very highly abstract or stylized ideas of what a basement membrane might look like. This part right here, that whatever color that is, the very pale orangey brown thing that says basal lamina there, that is the basement membrane. This particular basement membrane is the one that exists in between your epidermis of your skin and then the papillary layer of the dermis, or the areal or connective tissue within the dermis that supports the epidermis of your skin. So this yellow line right here is the plasma membrane. Here you have a desmosome, half of one in this case, so they call it a hemidesmosome, which hopefully you're familiar with from chapter 3, cells. So these are very strong proteinaceous structural attachment points for cells. And in this case, the internal apparatus of the desmosome is connected to these green proteins, which are called integrins or integrins, and they protrude through the plasma membrane and attach to the basal lamina. So all of this stuff is the cytoskeleton of the cell, which is then riveted or bolted to this underlying basal lamina or basement membrane. And then if you look over here, this basement membrane is just an assortment of other proteins. You don't need to know the names of all of them, but they're basically all woven together. I don't know if you can see, there's sort of this grid of this red protein. So you can think of it as like a loosely woven fabric or fiber, only it's made out of protein instead of something like polyester, and it's used to separate tissue layers, quite often separating epithelia from the connective tissue that are underneath them. And the basement membrane will come up again when we do skin, so I just wanted to point it out there. And then epithelia also tend to be avascular, which means they do not have vascular tissue or blood vessels. So they are going to rely on the connective tissue underneath them for nutrient and gas exchange and exchange of waste products as well. Innervated means that epithelia can have nervous tissue embedded within it. If you will remember, or you will learn when we do skin, if you haven't done skin yet, that there are Merkel discs or tactile cells that exist within the epithelium of the skin. Also, one thing that many epithelia, or I should say all epithelia, have in common is that they're easily regenerated. And this makes sense, right? Because the epithelia are covering all of the surfaces. Surfaces have a tendency to get damaged, and so they need to be rebuilt on a pretty regular basis. Especially think about your skin. It gets scratched all the time. Skin cells get old and dry and die off, so they tend to be always being replaced or easily replaceable. Let's see, what's next? For lab, I just threw a couple of lab slides in just to give you a heads up on how we're going to cover these tissues throughout the course of this semester. So anytime we introduce a tissue like an epithelium or a connective tissue, there are things that you're going to want to know about that epithelium or connective tissue. You are going to want to be familiar with its description. More importantly than that, you are going to want to know its function. So anytime an epithelium or connective tissue, a new tissue type, is introduced in lab, make sure you know the function, and then be able to give the location of that tissue as it relates to the organ system that we're covering that week, and you might even want to know where else that tissue type might show up outside of the organ system that we are talking about at that particular point in time. Case in point, this is stratified squamous epithelium. It is what makes the epidermis of your skin. You can also find it lining the inside of your mouth, and it is what makes up the epidermis or epithelium of your esophagus. Next up, epithelia. I forgot about covering versus glandular epithelia. Let's not really worry about that. And let's not really worry about this slide. And this we talk about in lab, so I will talk about very, very briefly. So you want to remember the difference between an endocrine gland and an exocrine gland. Endocrine glands do not have ducts, so there is no common pore or tube into which the secretions are secreted. They just tend to make it into the bloodstream, and then they circulate throughout the body. So endocrine secretions also tend to stay inside the body. Exocrine glands have ducts, so there is a pore or a tube of some sort that the secretion empties into, and then that secretion then either empties onto a body surface or into one of the body cavities. So you're thinking sweat glands or salivary glands. Either way, a body cavity, or I should say the body cavities that the exocrine glands empty into, also open or empty into the outside world. So your saliva is on its way out of the body as it travels through your digestive tract. Let's see, connective tissue we do have to talk about a little bit. So connective tissue is defined as cells suspended in an extracellular matrix. The matrix is then broken down into protein fibers and ground substance. So proteins you're probably familiar with. You want to think of them as very long rope-like proteins or rope-like molecules that are used to make things tough and connect things together. The ground substance can vary very widely from one connective tissue to another. So if the ground substance is minerals, you have a connective tissue called bone. So here you have a very tough mineral-like ground substance that has some collagen protein fibers embedded within it, and then osteocytes, which are not labeled here, but where this says lacuna, these little black things, that is where the osteocytes are. So you have cells suspended in this extracellular matrix of minerals and proteins. If you go all the way to the other end of the spectrum and your ground substance is a liquid, then you have blood as a connective tissue. And there are protein fibers floating around in this liquid. They are just dissolved so you don't see them. So you still have extracellular matrix being composed of protein and ground substance in your blood as a connective tissue. Connective tissues, because they are all so different in terms of their structure, do not have a lot of major functions in common, so we're not going to worry about that. The 4 different classes we'll talk about in lab as we introduce the different connective tissues, so we're also not going to spend much time talking about this slide. This slide is a little bit more important. So the connective tissues do vary quite a bit, but they do have some things in common. Numbers 1 and 2 being the most important. So they both have the same arrangement of cells that are suspended in, or as this says here, separated by a non-living or non-cellular extracellular matrix. The other important thing is that all of your connective tissues, from bone to blood and everything in between, is derived from an embryonic tissue that's called mesenchyme. So even though blood and bone don't look or act anything alike, because they share the same embryonic derivative or the same embryonic tissue, they get classified together as connective tissue because they're distantly related embryonic cousins. The other 3 and 4, some of them are vascular, some of them not so vascular. Some of your connective tissues are innervated, some of them not so innervated, and then also their regenerative capacities or capabilities are also highly variable. So there isn't a lot to say about 3 and 4 other than the tissues vary a lot in their degree of blood vessel vascularity, whether or not they're innervated, do they have sensation or not, and how well they're regenerated. Just to give you an example, cartilage doesn't regenerate very easily. If anybody's ever damaged their meniscus or started to develop arthritis, you know cartilage doesn't grow back very easily. Blood, on the other hand, you are always replacing. So blood is a connective tissue that is easily regenerated. And for lab, I don't want to run out of time here. It looks like we're going to go over 15 minutes and just going to have to be okay with that. Lab, what do we want to know about lab? We are going to go over different connective tissues in lab. You want to be able to identify them when you see them. You want to know which cell types are associated with that connective tissue, which fiber types, and then the function of each connective tissue and examples of where you find it within the body. Let's see, the nervous tissue we don't care about. We have a whole chapter on that. Muscle tissue, we'll have a whole chapter on that. Alright, so we are going to stop here because we're just about out of time, and we will pick up membranes in the next video. That one I promise will be much shorter. So see you in the next video. Bye.